Hello. This is a continuation of the first lecture in the series of lectures for the lab that I'm giving on tissue. And as you know, since you've looked at it already, the first presentation was about epithelial tissue. And this presentation is designed to reinforce that presentation, except that we are going to have visuals instead of just me writing out notes. So this is a PowerPoint presentation, and as I go through it, we'll be talking about epithelial tissue visually. Um, so let's go ahead and let me start here. And I'm going to do that, and I'm going to say next slide. So we're going to start off with simple squamous epithelium, and we're going to talk about what it looks like. We'll talk a little bit about its structure and so forth and on. So this is a slide that shows us simple squamous epithelium. Get rid of that. I don't really want that. And you can see two different areas that are being identified. Here, this area, okay, that area right there, and these cells right here. Those are simple squamous epithelial cells, and this is taken in the lung, and that's what they would look at. So this is a view of epithelial tissue from the side. So if you're thinking about a plate, remember simple squamous epithelium, a single cell layer thick, and it's flat cells. So if you're looking at this, if you visualize this as a plate, each cell being a plate, what you're looking at is from the side of the plate like this rather than down from the top. And let's go to the next slide. So here is a nice little slide that shows us, again, the simple squamous epithelium. And remember, we always have a backing of basement membrane. And in this case, I added this slide because we've been talking about serous membranes quite a bit. And here is an example of where we see it. So in the lung, so we have the visceral pleura and the parietal pleura. They both are constructed in this manner with simple squamous epithelium and a thin layer of basement membrane. And so we can see that this is exactly simple squamous epithelium. In this case, it's the serous membrane. So I thought this slide was interesting just so that you can connect that idea, serous membrane, to the epithelial tissue and see it also, again, reminding yourself of what that's all about. So going to the next slide. So here is a nice example again. We have flat cells with a very thin basement membrane, simple squamous epithelium. Here we have a view of simple squamous epithelium, but now we're in a blood vessel. So if you look closely, these are blood cells here, and the lining or the wall of the blood vessel is simple squamous epithelium. So we have here, there is a cell. That's simple squamous epithelium. So if you recall, lining and covering epithelium lines body surfaces and body cavities, and empty cavities such as a blood vessel are also lined with simple squamous epithelium. So here again, another example. Notice the terminology, mesothelium. I'm going to talk about that in a little bit, but just keep that in mind. There's some terminology here that might get you confused. We're going to talk about and straighten that out, and I'll talk about it in much more detail in the lecture. I will cover it a little bit here in this presentation. Again, we have our simple squamous epithelium here, here, and here. So those are all examples of simple squamous epithelium. You're going to see that, and I apologize for this slide. It's terrible. I tried to do this freehand. Not good. So, but what we're looking at is some simple squamous epithelium. Again, here's a nice one right here. Let's just outline that one right there. Okay. This is your lung. So this is a image that I took through a microscope that I have. and this is a cross section of the lungs. So this is also showing your respiratory membrane. You're not seen in the image. They're too small to be actually identified. There are blood vessels, capillaries that are infusing this whole area, and air is moving. If there's, can't see it, but there'd be a little blood vessel right here, and air would be moving back and forth, or I should say, oxygen would be moving in. I'm sorry, from this area into the capillary, and then carbon dioxide would be moving out into this space here. So this would have oxygen in it here, and carbon dioxide. What? Crazy man. Okay, carbon dioxide, let's do it this way. Yeah, getting used to this. CO2. Oh, this is so much fun. There you go. All right, so there, oxygen and carbon dioxide would be in these cells. Then over the edge of these cells is what you would get the exchange of these gases. Okay. All right, so here, this is from your textbook. If you're in the chapter 3, again, just emphasizing the fact that here we are, simple squamous epithelium. And we're going to look at this writing here in the next slide, and we'll walk ourselves through that. Okay, so simple squamous epithelium, a single layer of flat cells. Resembles a tiled floor when viewed from above the apical surface, and a centrally located flattened or oval spherical in shape. Okay, so centrally located nucleus. Now here's that terminology I was talking about. Endothelium and mesothelium. So what do those words mean? They are referring back to epithelium. Endothelium, endo meaning within, so a covering within. Mesothelium, a covering in the middle, basically. So endothelium is type of cell, you would find that in lining blood vessels, and we saw that earlier. Mesothelium, you would find those cells lining body cavities, such as the thoracic, or better still, the abdominal pelvic cavity. It also refers to the germinal layer from which this epithelial tissue arises. Now, we haven't talked about that, but we are going to talk about that, and I will explain what that means, germinal layers. But so don't get confused by the terminology. Endothelium, mesothelium, you're still talking about epithelial tissue, even though you don't actually say epithelium, you say endothelium or mesothelium. So let's talk a little bit about function. So function is filtration, diffusion, and secretion, particularly when we're talking about the serous membrane. Okay, it is not found in body areas subject to mechanical stress, such as wear and tear. Okay, so what you're going to need to do is be able to identify where. So you're going to need to know location on the test. You're going to need to know function on the test. And these tables are going to give you that information. Okay, so let's move on to the next. So here now we're looking at a single layer of flat cells, simple squamous epithelium. And let's go ahead and I'm going to outline one of those cells. Here they are. Okay. So that's a single cell. Here's the nucleus. And you can see them all over. It's like a mosaic, a very chaotic mosaic. Now, remember when I talked about in lecture that you're looking down from the top and you're saying to yourself, I don't know if that's multiple layers or single layers. And I told you that, well, if it were a multiple layer, you would see ghost-like looking nuclei underneath that. And if you look up here, for example, you're looking at a little ghost-like nucleus here and a dark one right there. And you might say to yourself, oh, that's what he's talking about. But that means that we have multiple cells. And yes, we do. But what happened was, when they made this slide, the very thin layer of skin, this is a frog skin, got folded when it was put down onto the surface of the slide. So some of it folded underneath, and so you're actually looking through and seeing the folded part underneath the original. So this is not actually multi-layered. It is in fact a single layer, but because of the way it was prepared, in the process of preparing it, they turned it into a multi-layered affair, and you can see the little ghosty nuclei under the first layer. But if this were prepared properly, you would not see that. Okay, so again, here we go. Same view. Here's one of the cells right there. Simple squamous epithelium. All right. And why is it dark in some areas and light in others? Just the way the stain took onto the surface of the frog skin. So that's, again, as a result of the technique in preparation. So here's that slide that was so terrible before. And as you can see, again, simple squamous epithelium, except now we're seeing it from the side rather from the top. This is a better prepared slide because we don't see cells underneath. We do, in fact, see a single layer, and there we are. That's one layer, and here's the nucleus. And you see that little dot right there? That little dot right there. And you can see it in a number of other places. Let's see if there's one right there. Okay, there's one right there. There's a little dark dot right there, and here's a nice one here. Okay, I'm pointing this out. Here's nice. These are nice right here and right here. Why am I pointing these out? These are the nucleoli that you learned about in cells. This is where ribosome construction is going on inside the nucleus. There's a nice one right there too. Okay, so those are all. That's all a good example of that structure that sometimes we find inside the cell nucleus. Okay, another example. I'm showing you multiple slides. Why am I showing you multiple slides? Because when you are taking the test, you're going to see this tissue in multiple different ways. And so what I'm hoping you get from this is a search image and the ability to understand the sort of the basic structure. So no matter how you see it, you'll be able to identify it. I guess the best way to talk about this is to say, well, okay, if I show you a car and I only show you one car, let's say a Mustang, and then I say this is a typical car, and I just show you the Mustang over and over again, you may not be able to say to yourself when you see a VW Bug or a Maserati that, oh, that's a car. So what I'm trying to do is show you all the different ways in which you can see the car so that you can see the basic premise of a car, what it looks like basically. So no matter what model you look at, you're going to identify that as a car. Okay, so there's one other thing in this drawing that I want to point out, and I'm going to find out where, there it is. So look at this. This is something in the frog skin. This is actually a opening to a gland. So you can see the gland in the background right here, and this little pattern right here, that's the opening to the surface. So the frog's skin is producing something. Here's in this corner, you can see here's the opening, and then here's the duct that leads down to some gland in the. So you can see it here too. Let's see, not there, but here. Where's my? There it is. So you can see it here, same structure. It's just interesting. You can sometimes see things that are not actually related to what we're talking about, but are interesting nevertheless. Okay, now simple cuboidal epithelium. Okay, so simple cuboidal. Let's break the words down. Simple, single layer, cuboidal. Simple layer, single layer of cube-shaped cells. Okay, all right. So here in this case, we're going to look at the kidney, and here they are. There's one there, cube-shaped cells. Okay, nice little image, cube-shaped cells right there. Okay, and look at that nucleus. Look at that big oval nucleus. It's taking up a lot of space inside that cell. This is typical for this type of tissue. You can see it all around here. This kind of reminds me of a bracelet, sort of, with little stones that encircle the wrist. That's kind of the way I look at it. Not a very pretty bracelet, but nevertheless, you get the idea. Okay, so here it is. Single layer of square or cuboidal shaped cells. You got your basement membrane. Okay. So also notice there's no blood vessels in here. Here's the blood vessels. They're very close, but not inside the epithelial tissue. And so this means that exchange has to be in this direction between these cells, okay? Excuse me. Something caught in my throat. All right, so this is the table 4.1 again. The information, let me go ahead and bring that up. Okay, so simple cuboidal epithelium, a single layer of cube-shaped cells, round, centrally located nucleus. Okay, that's going to help you find that and see that. So where is it find? Where you find it? So it covers the surface of the ovary, lines and anterior surface of the capsule of the eyes, pigmented epithelial layer of the posterior surface of the retina, kidneys, kidney tubules. Why do you keep coming up? Smaller ducts, mini glands, thyroid gland. We'll look at that. There's an image of that. And then and also the pancreas. Function, secretion and absorption. Secretion and absorption. Okay. So here we go. There it is, the epithelial cells. There is the connection between, or the wall between, I shouldn't say wall, but the membrane that separates one cell from another. And here I did a little bit better job in drawing these black lines this time. Again, cuboidal shaped cells. So all of this you don't have to worry about. It's these structures right here and here, okay, and here. Oops, getting used to this. There, okay, and over here, these here, here, and there. Those are all examples of the ducts inside a kidney, and they're lined with the cuboidal shaped cells. Okay, all right. Now here's another image. It's a little bit harder to see, and you can't see really very well there. Let's go down. There you go. A little bit. Okay, so again, once again, I'm showing you this because you're going to see many different views of this, and we need to be able to identify and recognize it for what it is. Here's something interesting. You see it's got a very fuzzy edge here. Not clear edge, not a clean edge. And why is it fuzzy? Particularly here. See how fuzzy that is? That's because the ends of these, or the apical surface of these cells, have microvilli. So the cell has got these little finger-like extensions at the end of it, and it makes for a fuzzy edge instead of a nice crisp edge. Okay. And that's why you see that fuzziness. Here again, and let's go ahead and identify one of these cells. There's a nice one right there, simple cuboidal epithelium. Here's a nice one right here, simple cuboidal epithelium. Also over here, a little bit more difficult to pick out, but these are, and you know, look at the side, look at that nucleus, big round, takes up almost all of the inside of that shape. This would also be, but we're not looking at it at the correct angle to really see the cell itself. But if you look over here, we can just barely make out that cell there, okay? So again, all of this is an example of simple cuboidal epithelium. Again, simple cuboidal epithelium here. Nice view of that, okay? Again, not exactly square, but close enough. Certainly not flat, and certainly not column-like. Again, we get sort of that big, round, oval nuclei in the middle of the cell there. Over here. Okay. Same thing. There it is. right there. Big, nice. Oh, look at there. Look at that. Look at that nucleolus right there. Very nice. And in fact, as you start looking around, you'll see some more. There's one there. There's one there. Okay. So look at nice. Oh, nice. There you go. Oh, look at that. Yeah. So exciting. There's a nice one right there. Nucleolus, inside the nucleus, where you're going to get ribosome construction. All right. So what is this? Or what are we looking at here? Again, we're looking at simple cuboidal epithelium. We see that characteristic shape of the nucleus. Characteristic shape of the nucleus. Okay. And if we can just use our imagination a little bit, we can picture that big, square, cuboidal shaped cell. Okay. This is the thyroid gland, by the way. So this is a gland, and it's producing thyroid hormones, and the chemistry is all taking place in here. So as you'll learn, what's happening is materials are coming into the cell produces a substance, secretes it into here, and in this space, some more chemistry happens, and then it's taken back up into the cell, put into the blood as a finished product, a hormone. Okay. Non-ciliated, simple columnar epithelium. Non-ciliated, simple columnar epithelium. Let's look at some examples, okay? All right, so here is, let's see if we can, there, oh, get out of here. Can we go back? I don't think so. I don't know how. So there it is. What can you do? So here it is. And there are the microvilli. That's why you have the fuzzy edge. I'll show you that. We also have these specialized cells right here. These are goblet cells. They're producing mucus. And we'll talk about that a little bit more in lecture. Okay. So here's that fuzzy edge. Why is that fuzzy? Because of the microvilli that you see here. Okay. All right. Non-ciliated, simple columnar epithelium. The structures that we referred to, microvilli and the goblet cells. Okay, so that's something you should be aware of. Locations, gastrointestinal tract, ducts of many glands, and the gallbladder. Function, secretion, absorption. So, and you can go through and look at the examples of where they're talking about secretion and absorption is taking place. All right, so this is a view of one of those cells we talked about. A goblet cell. It is part of the epithelial tissue. It's a modified columnar epithelial cell, and what it's doing is it's producing mucus, and you can see here the sort of the overall structure. You can see a lot of smooth endoplasmic reticulum, which makes sense. The nuclei, ribosomes, and this is going to be the mucus here. All right, so first of all, let's see if we can identify one of these cells. And we're going to do that. Okay, there's the cell. Notice now the difference in the shape and position of the nucleus. So, God bless America. All right, so let's do that again. Here we go. There is, get away. There you go. Nucleus. Okay. And notice that there's this space here. If I draw a line along this edge, we have all this space. This is all part of the cell. This is the lumen here. And, oh, I'm getting fairly good at this. Okay. Fuzzy edge. What does that mean? That's the microvilli. But coming back to my discussion with the nuclei, notice that they're positioned at the base of the cell. They're elongated ovals. And there is all this space above this, starting to get upset. All this space above the cell, above the nuclei, before you actually leave the cell. So that's something you should be aware of. You should be able to identify that. Okay, so let's come back to this image. So here we go. This nuclei belongs to a different cell. There's my oval shaped. Again, you see, we can draw a line across here. We've got all this space between here and here. And this would be the bottom or the basal surface of the cell. Okay. Again, we get the fuzzy edge. That's the microvilli. And this structure right here, that's a goblet cell. Okay. Here, over here, goblet cell. This is actually the mucus, the sac of mucus inside the goblet cell. So typically that's what you're going to see. It's not always easy to see the cell itself. Again, a little more difficult here to actually pick out a cell and outline it. If I were to do that, it would probably be something like this. But it doesn't matter, does it? Because I get this pattern. Here's the edge of the nuclei. Here's the shape, elongated oval. Here's the basal surface. And we get all this space right here, okay, before we get to the outer edge of the cell. And we still have, this is a nice picture of the mucus. In fact, all of this stuff here, that's mucus. That's been released by these cells and is in the lumen of, this would be the small intestine. Okay. Same thing. Here is the apical surface. Here is the basal surface. Here are my nuclei. Here is that space I keep talking about. Okay. And here we also have our goblet cells here. So you can see them. Here's a nice, this is actually a decent, right there, nice goblet cell right there. Okay. This is all connective tissue in here. We'll talk, you'll learn more about that when you get into the digestive system. All right, now, where are we? What are we looking at here? Again, long column-like cells, okay? I can draw a line here and draw a line here, and that basically encapsulate where are the nuclei. I have all this space out here. That's free space inside the cell. Same thing, along here, okay? So this is the outer edge of that cell. I've got all this space here. Then that's going to tell me I'm looking at simple columnar epithelium. So this is a finger-like structure inside the small intestine. And it's not a microvilli. It's literally a folding of the layer of the surface of the small intestine. And these are ducts. You'll learn about these. These are lacteal inside that structure. And it's a nice image of that right there. Here's one wall. There's the other wall. Okay. So that's a duct that passes through the center of this finger-like structure. And this is where you're absorbing meal. So the meal out here is being absorbed into these cells, and then most of it goes into the blood. But some molecules, such as fatty acids, are too large to be transported into the blood, and so they get collected here in this duct, the lacteal, which is part of the lymphatic system, and then eventually return back to the blood. You'll learn about that in the digestive system. Stratified squamous epithelium. Okay, so again, we've got these layers of flat cells, and so this is just an overall picture. They're using the vagina as an example. Let's move on. Okay, here's a nice image. The flat cells. This is in black and white, obviously. These are the flat cells. These cells don't look particularly flat. These look flat. Why is it called simple squamous epithelium then, or stratified squamous epithelium? Because this is my functional layer right here. That's where I get the function out of this tissue. Down here, these cells are dividing, they're maturing, they're going through a process of development, and as they go up to this surface, they're dying out and becoming flatter. Here it is in color, same image. And we get the same effect. So the same, we get the more or less almost column-like cells here actually. And then as we move up in this direction, the cells are slowly changing. They're becoming flatter and flatter. Eventually they'll die, and at the surface then all we have is dead flat cells. Okay. Okay, let's go. All right, so stratified squamous epithelium. Let's see, what else can we see? So I'm going to ignore a little bit this keratinized stratified squamous epithelium as opposed to non-keratinized squamous epithelium. We'll talk a little bit about that more later on. Very briefly, this keratinized stratified squamous epithelium has this protein called keratin in it. Right? The non-keratinized stratified squamous epithelium does not contain keratin. So this is mostly the skin. Okay, so the skin is keratinized. Okay. When we find stratified squamous epithelium in other places, like inside the body, the mouth, for example, or the vagina as they used in the past, it's going to be non-keratinized type. Okay. All right. It is also producing mucus, so keep that in mind. Even though you have many, many layers of flat cells, does not mean that it cannot produce a mucus and put it onto the surface of that tissue. Okay, so location. Keratinized variety forms of the skin. Non-keratinized, we find it lining the mouth, the esophagus, parts of the epiglottis, the pharynx, vagina, right, and covers the tongue. What is this all about? Protection, protection, and protection. That's the big thing right there. Protection. Protection. So keep that in mind. It's all about protection. All right, so I'm getting better at this drawing, but you can see here, this is the edge. And we don't get a nice clean edge here. We get this sort of in and out, finger-like valleys and hills. That's because it increases surface area of the stratified squamous epithelium to connect it to the underlying connective tissue stronger. The more surface area, the stronger the connection between the connective tissue and the epithelial tissue. What's all of this stuff here? Why do we have these spaces here? This is an artifact. So all these little spaces that you see are artifacts. Here and even here you can see these spaces. So when this tissue was prepared, we had a block of this epithelial tissue, and we used a special tool called a microtome to slice that into very thin sections. So when it was put on the cover on the slide, we could look through it. Light would pass through it, and when the knife passed through, it caught the edge of the epithelial tissue and pulled it away, and created these spaces that you see here. So all of that is artifact. Okay, so here it is. This is the edge. Okay. This is where my basement membrane would be. Okay. And here's the outer surface. And we get this great big thick layer of tissue. That's what you're going to look for, is that thickness of tissue as you go through and look at slides. And that's going to tell you that that is stratified squamous epithelium. Again, here is the bottom layer. And you can see here the cells are not particularly flat, but by the time you get to the top, we do have flat cells. Okay? So the cells are not as distinct and nicely lined up as some of those other things. But again, I have this thickness here. And that's going to tell me, this thickness is going to say, stratified squamous epithelium. And there it is. Okay? So stratified squamous epithelium. Same thing. Here's the basal surface. Here's the outer surface, the apical surface. From here to here is my stratified squamous epithelium. So I get this thick covering of tissue. Sometimes, not always, but sometimes you can actually see that these cells are not flat. They're more or less round or cuboidal. But by the time you get to the top, you see that it's got this kind of flattish appearance up here. And so when you see this very broad band and this pattern, you know that you're talking about stratified squamous epithelium. Okay? Where is the stratified squamous epithelium? Here? Here? No. There's the edge. Here's the apical surface, and there's my band, and that is stratified squamous epithelium. This is muscle tissue, and this looks like glandular tissue, actually. I'm not sure what this is. I don't have enough resolution there, but this is definitely muscle tissue here. And then this is the rest of this yellow stuff is connective tissue. Yeah. So this is, again, an example of stratified squamous epithelium. The big band, or the thickness of that band is what you're going to be looking at. Again, same thing here. There's my basal surface, apical surface. I've got this big space, dark band. Muscle tissue, connective tissue, connective tissue. And this is, I can see that that is also muscle tissue. Same story. There is the basal surface. There is the outer edge. And there is my band, thick band. Okay, and here there is no real way to see the distinct cells. So what you're going to have to depend on is this pattern. This dark edge, and then that edge, and that thickness. Stratified squamous epithelium. Transitional epithelium. So what is that? Well, that's type of epithelial tissue that transitions from one shape to another. So we had flat cells, cuboidal shaped cells, column-like cells, and then transitional cells. And they are literally transitioning from one shape to the other. I'm going to kill this thing. There you go. All right, so how are you going to tell what this is? I'm going to get to that in a second. I'll show you. But here is the basic pattern. So there is the cell there. And I'll show you the basic pattern here. So there is the cell there. If you look down here, they're smaller. Not as easily seen as in this picture, as we'll see a little later on. But so the pattern will be small cells down here, and as you get up here, the larger their cells are turn up to be larger. And there's a good reason why that's the case. And this is where we're going to find it. Most of it. In fact, I can't think of any other place. Well, there's some in the urethra. There's some in the urethra, but most of it is going to be lining the urinary bladder. Okay. So here's what happens. So you get this. Here is the relaxed bladder. It is not filled with urine. And you get that cells on the surface in particular are nice and round, are almost cuboidal in shape. And then when you fill it and you have that full bladder, you need to go to the bathroom, the cell is flattening out here. Right? And that's to create a larger surface area, because this is going to expand like a balloon. And that surface that lines the urinary bladder has to stretch. And this is how we're doing it, by taking cells which are capable of stretching and transitioning between sort of a roundish, almost cuboidal shaped cell to a flatter cell here. Okay. Also notice that the thickness diminishes, because as the cells flatten out, this distance is reduced. This distance increases. And so you get the expansion of the outer surface of the urinary bladder. Now, this outer edge is expanding more than this inner edge. In other words, this has to stretch a further distance in this direction than here. And so the cells up here start off as a larger type of cell. You can see it here, a larger cell here than down here, because these are the ones that are get to stretch the most at the upper surface. So, again, you get the larger cells here, you get the smaller cells down here. And when you stretch this out, this collapses and it becomes more linear and flattens out. Okay. Okay. All right, so Why did they do this? I did not see this. Pretend like that's not there. Let's move on. All right, so here we go. There. And smaller down here. Gosh. Nuclei, scattered nuclei. I think the only way you're going to get confused here is if you think this is some sort of stratified cuboidal epithelium. First of all, little inside information. I'm not going to show you stratified cuboidal epithelium. There's just not enough of it in the human body to really have you see that and memorize that. You will run into it, talk about it, and lecture a little bit, but that's about it. You're not going to, if I'm showing you an image, it's not going to, I'm not trying to show you stratified cuboidal epithelium. You can see that it is stratified because we have the nuclei that seem to be stacked on top of each other. But if you look at the shapes of the cells from top to bottom, you'll notice a difference. Larger up here, smaller up there. Let's take a look at. All right, so smaller down here, larger cells up here. Okay. It does look stratified, but the cell shape is not, you can't really describe that as cuboidal. Can't really describe that as columnar either. Certainly not flat. So you get all the stacked cells and no really definitional shape that you can identify readily. But you do notice that they are larger up here at the edge than they are down here at the basal surface. And so you're saying to yourself, okay, that's stratified, that's a transitional epithelium. Okay, same thing. They get larger up here and smaller down here. Okay. This is connective tissue here. Same pattern, larger up here, smaller. And the nuclei are more densely packed down here because the cells are smaller. So the nuclei are closer together. Out here, because the cells are bigger, they're more spread out. Same thing, a lot of cells down here, a lot of nuclei here. This is the basal surface here, and this is the apical surface. Notice how much larger they are here. The nuclei are spread out from each other, and they're not so densely packed together. Here they are, fairly densely packed together. Same thing. Look at this is dark here because the nuclei are very closely packed together, and the nuclei are picking up that stain a lot more than the rest of the cell. And so this tends to be darker down here, and this is lighter up here. You can't actually see the cell shape very clearly, but you can see we have many cells. We have a thick band. So you're saying, oh, because that's stratified squamous? No, because we have the pattern of nuclei is different than what we saw in stratified squamous. First of all, by the time you get to the outer surface of stratified squamous, you're not going to have any nuclei. They're going to be gone because these are dead cells. And we get instead a lack of nuclei at the outer edge. But here, the nuclei are found from top to bottom. More densely packed here, but still present in the upper area. Okay. So same pattern. A lot of densely packed nuclei, not so densely packed. Cells are larger here and smaller down here. Okay. All right, ladies and gentlemen, that ends the presentation for epithelial tissue, and I will see you in the next lecture.